Katie, are you there? Katie, we can't hear you. Sorry, everyone, just bear with us. We're going to try to sort out Katie's audio issue. While we work through that, I was going to run a poll at the very end of the webinar. I'm going to take the time now to launch that poll. And so what we're hoping to do is, through answering some of these questions, is learn a little bit more about the work that you're doing. So are you currently using any imaging in your work? So there's optical imaging. If there's other imaging that you're using, it would be really great to learn more about the imaging that you're using, and you can enter that in the chat window if you'd like. Perhaps that you're not using any type of imaging, but you're looking to move to using that imaging in the future. And Katie, if you want to try talking, I think we can hear you. Katie, did you want to try talking? I think we can hear you. I'm going to give everyone a few more minutes just to respond to the poll. The second question is, following the webinar, obviously you haven't listened to it yet, but would you like us to reach out more to talk about your specific needs for the Newton system? Or are you just here to learn more about different technologies? Again, also, we're going to record the webinar, so we can send that link out to you as well. Katie, I think we can hear you.
Hey, Tanya, can you hear me?
I can, yes.
Beautiful.
Okay, perfect. I'm going to end the poll question now, and then I'm going to pass things over to Katie. All right, Katie, it's all you.
Awesome. Thanks, Tanya, for the introduction. And welcome to everyone that is joining us this morning. So as Tanya mentioned, today's webinar is going to be a little different for those of you who have tuned into some of our previous webinars. It's not going to be product focused. So today the goal is more of an educational session where I'm going to walk you through some of the common obstacles and more so the how-to style questions that often come up during optical imaging sessions. Okay, so today's topics of discussion, first I'm going to start off just by reviewing optical imaging in general. So this may be a review for some of you that are already using optical imaging, but I want to make sure that everyone is on the same page as we dive into some of the more extensive questions. We'll talk about how to add optical imaging to your lab. So this can be useful for those that are just starting to learn about optical imaging and may have concerns about what that looks like in terms of the infrastructure and costs. We'll talk about the considerations when designing your animal experiments, and then we'll finish by talking about how to get the most out of your imaging data. Okay, so to start off, just to review optical imaging. So this is confusing for those that are just jumping into the field, as optical imaging is actually quite a broad term. So it can actually be broken down into fluorescence and bioluminescence in terms of animal imaging studies. So for fluorescence imaging, the administration of a fluorescent imaging agent, so either a fluorophore or a fluorescent protein, happens, and then an excitation light of appropriate wavelength is used to illuminate the subject. In the case of using a protein such as GFP, you'd have to first engineer the cell line of interest to express the GFP, or you could purchase, for example, a transgenic animal that already expresses the reporter gene of interest, and then you would illuminate the subject. And we'll talk a little bit about this in the coming slides. Now, this would lead to excitation of the fluorophore or fluorescent protein, and the subsequent emission of light. And then that light gets detected by the CCD camera above, which you see here. And then the signal gets analyzed and converted into an image that would detail the location of emitted light from the subject. Now, bioluminescence imaging is very similar. The animal model first needs to be developed such that it's capable of expressing or secreting a luciferase enzyme. So again, this could be a transgenic mouse or a mouse that's been injected with cells that can either express or excrete luciferase. Then the main difference here is that the appropriate substrate needs to be administered to the animal. And then this is what leads to the production of light. So it's that enzymatic reaction of the luciferase with its substrate. And then similarly to fluorescence, the light is then detected by the cooled CCD camera and the image is produced. Now, unlike fluorescence imaging, bioluminescence doesn't need an external light source. So due to this, and because tissues don't exhibit any endogenous bioluminescence, some people prefer bioluminescence since greater sensitivities and higher signal to background ratios can be achieved compared to fluorescence. So just briefly touching on, the common applications. This is, of course, not the only applications. The possibilities are really quite endless, but I would say these are probably the most commonly used applications in the field right now. So cancer studies for sure. Many people use optical imaging both to track cancer cells, also looking at treatment studies for cancer. Neurology is a big one, especially for bioluminescence imaging, since that's a bit more sensitive than fluorescence, and you can detect signal deep within the brain. Biodistribution studies is a big one, since optical, nicely shows the whole body imaging, which we can't always get with other modalities, or it can be quite expensive to do so. Treatment studies, as I mentioned, and this is, I would say, more specific to bioluminescence imaging, since for a lot of the luciferases, you need cofactors, such as ATP and oxygen, which we'll talk a bit more about later. And that is important for treatment studies, because then you can get things such as viable tumor tissue, for example. Cell tracking studies is a big one. And then finally, ex vivo imaging is becoming more and more popular, both for fluorescence and bioluminescence imaging. Okay, so moving on to how to add optical imaging to your lab. So this is a really important topic that I wanted to cover, because I think there's a lot of people out there that are under the impression imaging can be really intimidating for a lot of groups that have, never really had imaging tools in their lab. And many are under the impression that you need, quite a bit of expertise to add imaging capabilities to your lab. Optical imaging is kind of a unique modality in the sense that really anyone can use it. And for the most part, these systems are very much plug and play. And we'll talk a bit more about this, but there's ways that you can adapt the workflow to really simplify things and make this a system that anybody can use. So first, what are the things that you need if you want to think about incorporating optical imaging into your lab? So there's not a lot, and you can make it as complicated as you want to. But first, you need a lab bench allowing animal work. And I would say not even necessarily that, because there's lots of people that just use optical imaging for in vitro or ex vivo studies. So maybe you don't have any need to do animal studies, but I would say you need a lab bench that is able to fit a small footprint system. So here I'm showing on the right, this is the Newton system. And as Tanya mentioned, this is the system we carry. But keep in mind, this is probably a fairly standard size for most optical systems these days. There are some that are bigger in terms of length, so they wouldn't fit on a benchtop. But for the most part, the 3 to 5 mouse systems are fairly small. It's just kind of a black box, which you do the imaging in, and then a computer that will allow you to do set the parameters and the analysis. So not a lot in terms of space needed. Secondly, you're going to need imaging substrate. And again, you don't necessarily need that if you're okay with just doing fluorescence imaging. But for bioluminescence imaging, you will need to consider that may be a consumable you will need as you progress through your studies. And then finally, you need cells expressing your relevant reporter or optical dyes. And again, this can be as complicated as you want it to be. And we'll talk a bit more about this on the next slide. Okay, so as I mentioned, there's kind of two different groups that I wanted to focus on. There's many of you that have never used optical imaging and maybe have never used any imaging modalities, and optical is kind of scary. And then there's others that have maybe used other imaging modalities but are new to optical, or maybe you've used optical for quite a long time and you just want to know what are the other things I can start to add into my experiments to get more information. So we'll go through each of these separately. If you're new to optical, in terms of the system, I always want people to consider X-ray or CT. So nowadays there's kind of two different types of systems. There's systems that are just optical, and then there's also combined systems. So this is becoming more and more popular. And when I say that, I mean you could have an optical X-ray system or an optical CT system. So for those of you that are more comfortable with optical, maybe you have a system in an imaging core, or it's a system that's going to be used by those with more imaging expertise. That may be something that's important to you. So that's something to consider, because as you add in these modalities such as X-ray or CT, you can get complementary information. And again, we'll talk about this a bit later in the presentation. So there is an added benefit of having a combined system. For those of you who don't need that extra information, maybe you're just interested in the optical data, and especially those that are new to optical, I always encourage them to really think about how much they need the X-ray or CT data, just because there's a lot of extra considerations that come with that combined system. So we need to keep in mind that X-ray and CT has a lot more infrastructure and safety requirements compared to just a standalone optical system. If you're new to optical, I always suggest starting with fluorescence or F-Luc BLI, so firefly luciferase BLI. The reason being, for fluorescence, you don't need substrates, so that'll limit your consumables. And it's also one less variable to worry about. So if you get any funky signal when you're just starting out, you don't have to worry about, did I make my substrate correctly? Did my injection work? With fluorescence, your cells are in there. As long as you know that they're expressing your fluorophore, you should be fairly certain what your signal is representing. In terms of bioluminescence, if you're set on getting into bioluminescence, it's still very simple. I recommend firefly luciferase for those who are less familiar with optical, just to get started, just because this is the most commonly used reporter gene, I would say. It's very well published. We know a lot about the kinetics and how it interacts with other substrates. And it's also very sensitive. So it's definitely a good place to start. There's also lots of options for those of you that may want to purchase your cell lines or your substrates already made for you. However, those that are more comfortable with optical, there's lots of different avenues you can explore here, and this field continues to grow very rapidly. So I encourage you to, dive into the literature. There's many different reporter genes now to choose from, as well as substrates. Okay, so discussing substrates. So if you're new to optical, this is something else you can do to really simplify things. There's actually pre-made substrates that you can purchase. There's quite a lot of options, especially for F-Luc BLI. I always think this is a great place to start. So ReadyJect is one example that I'm familiar with, but there's quite a few options. And I think this is a great place to start, as I mentioned, just because this limits the variability. So once you get more comfortable, you can start to buy you can buy the substrate in the powder form and start to make your aliquots in lab. And that will save you a bit of money. You'll also be able to buy it in much larger portions. So that is a good option down the road. But initially, to start out, I think the pre-made substrate is the way to go. The concentration you have in your hands, it's aliquoted. So you can basically just take it out 5 minutes before you're ready to image, thaw and you're ready to inject. So that's really handy. For those of you that are comfortable with optical, there's also many new substrates that are being made for enhanced sensitivity. So that's something to look into. If you're new to optical, you also can actually order cell lines. So this is really ramping up. This used to be harder to do, I would say. There used to be probably a couple F-Luke cell lines that were fairly well characterized, but now there's quite a few options for groups that don't have the infrastructure or the expertise to engineer their own cell lines. For those that are comfortable with optical and either have the infrastructure to make your own cell lines, or maybe you have a lab that can collaborate with you and you can make them within your institute, that really opens the door for you to, incorporate any reporter gene that you're interested in testing. So that's really great for those that have the capability to do that. Okay, so I mentioned you don't have to have the expertise or infrastructure to engineer the cells. So this previously was quite a large obstacle for people looking to get into optical imaging. One resource I wanted to point out is ATCC. So for those of you in the cancer field, you are probably already very familiar with ATCC, as many of us have been purchasing our authenticated cell lines from there for years. They now offer many, many options for optical imaging. So they have luciferase labeled cells, they have the isogenic cells, and they have other reporter labeled cells. So cells expressing GFP or RFP if you don't want to get into the bioluminescence. I've also included a couple other options here on the right, and these are just a few that I know of. So feel free to explore this. I think this is a really great resource. And for the most part, these companies that sell these cell lines have already gone through and characterized these cells for you. So they'll actually show you the data of the expression levels and the cell to signal correlations. So that can be really handy. And that basically allows you to just move directly into your in vivo studies. Okay, now we're going to move into considerations when designing your experiments. So how long to image for? So this is a common question we get quite a bit. I always encourage people to run a pilot study. This is super, super important. The reason being, the timeline is going to differ between nearly every variable you throw into your study. So between disease models, between the strain of mice, even within that strain of mice, maybe you have 5 or 6 animals within a study, you're going to have variability between subjects. You're also going to have variability between time points. So as that disease model progresses, maybe it's an aggressive tumor model, your substrate availability is going to change. And so the kinetics of your signal is also going to change. If you absolutely cannot run a pilot, or you're just going in kind of blind, I would always suggest accounting for about half hour per mouse if you're using Firefly luciferase. So it's typically between, I would say, 20 and 35 minutes that you're getting peak signal. But again, keep in mind it's not always ideal just to guess, because you could account for half hour and it may not peak for 45 minutes. So always try and run a pilot if you can. And set up a sequence acquisition if possible. So for those of you that are familiar with optical imaging and already have it set up in your lab, regardless of the system you're using, your software should allow you to set up either, it may be called a sequence or serial imaging session, where you're setting up multiple images with the same parameters over time. What you can do is draw an ROI on that first image for your area of interest. And then you typically right click and copy that ROI and paste that exact ROI on each subsequent image, so that you're looking at the signal over time in the exact same area. Image as frequently as needed until the peak signal is clearly passed. So you can see I'm showing these figures on the bottom because there's lots of things to consider, and one of them is the substrate delivery. So depending on both the luciferase and the substrate you're using and how that substrate is being delivered. So for example, whether it's an IP injection or an IV injection, your peak signal is going to look very different. So I would say image as often as you can until you're confident that the peak signal is clearly passed. So some people get very tempted if you have a sequence set up, the first image where it looks like the signal is going down, they'll finish the imaging session. And in reality, if you kept imaging, you'll find it kind of goes in waves. So realistically, you really should try and take 2 or 3 images after when you see the peak starting to drop, just to confirm that what you think is the peak is actually slowing down. Okay. Again, pilot study is super important here. Consider how aggressive your disease model is. So this is really important in terms of how often should you image. For those of you using metastatic cancer cell lines, you'll know what I mean. So sometimes if you're using something really aggressive, it doesn't have to be cancer, but maybe just an aggressive disease model, your whole study time frame may be 2 weeks total. And so you need to fit your timelines or your imaging time points within that 2-week time frame. Otherwise, you're going to run out of time very quickly. Versus somebody that may be doing a PDX model. Some of the PDX models, because they're consistent with the patient timelines, they may take months, for you to develop a palpable tumor. And so in that case, you're not going to want to be imaging every 3 days. Your PI is going to be very upset. You'll be using a lot of substrate and a lot of imaging time. So I would say think about how aggressive your disease model is. Think about the total timeline in its entirety and how many time points you want to get out of that. Image as frequently as you can, but only if you're gaining new information. So I typically see kind of 2 extremes, people that are just imaging at one time point, and you're not really getting a lot of information from that. It's more just a static snapshot of your animal model. Or I see people that want to image as much as they can, and they're imaging every other day, and maybe it's a slow disease model. So it's really unnecessary. You're not going to see anything new potentially. So again, I would say image as frequently as you can, but only if you're gaining new information. If you're looking at part of a treatment study, choose your time points accordingly. So for example, you want to image directly before starting the drug to get the true pre-treatment measurement. So if we look at this image on the right, we see on the top, they are imaging at day 5, and that would be pre-drug, and then 7 days post-drug. So that means that they've imaged on the day that they start treatment. So I would say, if possible, always try and get your pre-drug image on the day prior to starting treatment. If not, at the very least, the day before, because you want basically as little time in between as possible so that you're getting a true pre-treatment measurement. If you need to image more than once a week, or if you would like to image more than once a week and you think that you can gain new information from that, I always suggest starting with a pre-injection image. And this can be confusing to people because, the substrate you shouldn't need to do what people call a pre-contrast image. However, keep in mind that because the substrate kinetics are going to be a little different, especially as your disease model progresses, you want to make sure that there's clearance of the substrate before you start imaging. And you'll be surprised if you start doing this. It's reported quite a bit in the literature that For example, D-luciferin only stays within the animal for about 24 hours. And so people assume you don't need to do this if you're imaging 2 days later. I think many people will be very surprised, especially for those of you that are doing aggressive cancer models, to see that once your animals get more sick and the cancer progresses, the animal will have a harder time clearing that substrate. And so you'll actually see that substrate, sticking around for 4 or 5 days sometimes. And so it's always important to start with a pre-injection image. Make sure there's no substrate sticking around from your previous imaging session before you're doing your next imaging. Will animal fur interfere with my signal? This is another question we get quite a bit. The short answer is yes. The good news is there's lots of things you can do to try and avoid this. So, of course, we always suggest using nude mice when possible, just to eliminate the fur entirely so you don't have to worry about this. Of course, that's not always possible. If it's not possible, white fur is better than dark fur, of course. You always have the option of shaving or narrowing the area of interest. So this is also quite a common strategy. I recommend for those of you that are interested in narrowing, and if you haven't done it before, if you get the sensitive, it's actually human nair, but for sensitive skin, it actually works quite well. And the animals handle it quite well. And typically it can be put into most animal protocols. So check with your institution, but this is quite common nowadays, and it's a pretty good strategy. It works quite well. Try to avoid animal models that have variability in skin tone. So this is actually something I wasn't aware of, and I discovered in one of my collaboration studies. I was doing some imaging for a group that was looking at melanoma, and they had an animal model with black fur. And so we waited until the day of imaging to narrow the area of interest. And what happened was, once we, got some of the fur off, we realized that underneath the fur, there was actually 3 different, colors of skin, kind of patchy. And that can really be a struggle, because that's all of those patches of different color are going to interfere with the signal differently. And so I would say try to avoid this if possible. I know it can be really hard if you're a melanoma group, and these are, the models that you're working with. If you absolutely can't avoid it, I would recommend narrowing early, even before inoculation, to try and see where the patchiness is and see if you can inoculate in an area that has something that's consistent, so that at least if you're getting interference with the pigmentation, it will be consistent in the area that you've inoculated those cells. And the most important thing I would say is to be consistent. So, whether you're using white fur or dark fur, whether you're shaving, narrowing, etc., just make sure that it's consistent, across your entire study, as well as over time. So don't just nair your animal before the first imaging time point and then image for 2 weeks. I would say you need to make sure that imaging area is consistently clear of fur throughout the entire study. So if you need to re-nair or reshave, try and do so that it's consistent. What else can interfere with my signal? Now, this is a good one. We don't actually get asked this often, but I think it's something that people miss. People tend to think of the fur. People don't tend to consider other things that can interfere with signal. And to be honest, there's a lot of things. So it's something good to think about. This is an example. This is actually a study I did in my PhD. And there's a lot here. So I don't want you to focus on the labels. I just want you to focus on the red images as we go through. So I'm going to start with the row on the top. And looking at the image on day 6, this is a mammary fat pad tumor. And you can see by day 13, we're going across the top row. So from day 6 to day 13, you see the tumor is growing. And as expected, the signal goes up. If we then look at day 19, again, this is the same mouse, the same tumor. The tumor has gotten significantly bigger. But you'll see for whatever reason, there's a dark hole. It's almost like a void of signal in the center of the tumor. What's happening here, we can see on day 20 from the bright field image. So I've highlighted in red the size of the tumor. So the tumor is huge. But what you can see in the center, in these mammary fat pads, this is quite common. They'll get like an ulceration or a scabbing at the surface of the tumor. And what ends up happening is this actually absorbs the light trying to get out. So you'll end up with this ring effect where you actually have loss of signal in the center and strong signal around that. You'll see the same thing in the second row. So we see an increase day 6 to day 13. Day 19, we almost see entirely an entire loss of signal in the center. In the bottom, you'll see this is a treated mouse. And so we'll see the signal, actually we'll start on day 20. The signal is much, much smaller. We don't see any scabbing because the tumor is not even palpable. And as a result, we see the strongest signal on day 19 is in the center of the tumor. So this is interesting. This is a common thing that happens, especially with the aggressive tumor types. So something to keep in mind. Other things to keep in mind are stitches. So I've seen that, especially with groups that are doing neurology studies. So it doesn't mean that you can't use stitches. I know many of you like to open up the brain for implanting your cells or maybe a treatment. You can still use stitches, but look into the literature and do your research and make sure that you're using something that's not going to interfere with your signal. Another thing is organs. Organs get in the way depending on where your signal is originating from. You have lots of things blocking signal. So we'll talk about that a bit more about ways around this. Okay, for those of you that are looking to detect very few cells, obviously we all would love to detect very few cells. There's lots of things you can do here. So there's many optimized high sensitivity reporter substrates. So as I said, the field is consistently growing. There's lots of options here for those that are interested. The injection site is important. So always try and inject the cells as shallow as possible. And that will limit the amount of tissue that's interfering with your signal. Use nude mice if possible. We talked about that. Position the area of interest as close to the camera as possible. So for most systems nowadays, the camera is going to be above the animal. And so if the camera is above the animal, let's say you're looking for lung signal, you're going to want to position the animal belly up. And so here I'm showing an example on the bottom right. If we're looking at a mammary fat pad tumor, for example, you're going to want to put the animal belly up so that the mammary fat pad tumor is surface level. Whereas if we're looking at brain signal, I would likely put the animal on its belly so that we're going through as little tissue as possible. Similarly, lungs or abdomen, likely you're going to put the animal on its back. You can also try to increase the binning. So this is essentially the grouping of pixels. So the higher the binning or larger the binning, you're going to improve your sensitivity. You can increase your exposure time. The longer you image, of course, the more light you can collect. I suggest starting with a single source of signal to simplify your results. So if you're just looking for, very few cells to start, I would say, injecting very surface level and don't do something that's systemic. So do put those cells all in one spot, whether it can be, the hind limb or the spine, something very shallow. And you don't have to worry about other signals within the body interfering. And then finally, you can adjust the field of view. So depending on the system, you can typically move the camera from above to zoom in on the animal, or you can move the stage itself to allow you to change the field of view and improve the sensitivity. Okay, and just finishing off, we will end with the section on getting the most out of your imaging data. So what units to report? This is very confusing. And that's because in the literature, people report both total flux versus average radiance. So it's important to note that both of these are widely accepted. And for the most part, you can choose to use either, and both will be appropriate. But there are obviously, there's probably a time and place that one makes more sense than the other. So for example, on the right here, if we look at the image on the right, where the animal has signal kind of all throughout, depending on what you want to measure, it might make more sense to look at total flux because there's signal all over the place. Maybe you're interested in whole body tumor burden. That would make more sense to use total flux versus the image on the left where you just have one tumor. You could trace around just that tumor rather than the whole body, and then you could look at average radiance. I would say, again, the important thing is to be consistent. So you don't want to switch the units that you're reporting halfway through the study. Make sure that you're consistent across animals and across time points. And if you're comparing signal over time, always copy and paste the same ROI. So this will be super important if you're doing total flux or average radiance to account for the area that is being measured. How do I know where the signal is coming from? So this can be really frustrating for many that are new to optical, but there are many options, so I'll go through a couple of them. The first is use optical to guide other complementary imaging techniques. So this could be something maybe like MRI, CT, or ultrasound. Here I'm showing an example of some data Tanya and I collected during our live demo in October, where we had some pretty confusing optical signal. Here it was kind of all over the place. This was an IP injection of ovarian cells, and it was very diffuse. But what we did was we used the optical to guide the ultrasound imaging as well as the MRI. And we are actually able to see in the end that the optical imaging really did make sense. We could see this tumor kind of going all the way through the upper abdomen here, as well as some tumors down here that were getting scattered in the lower part of the body. For those that are just doing a simple tumor model, it can also be advantageous to show multiple modalities. So here we have a mammary fat pad tumor with optical. So in the case of bioluminescence imaging, you can get a measurement of just viable tumor tissue. And then with something that's more anatomical, such as ultrasound or MRI, we can actually pull volume measurements out from this data. This is a study I did very early on in my PhD. And I just wanted to show this because I think many people see, when I say complementary modalities, especially for cancer studies, people think of tumor burden measurements. But I wanted to show this because it can actually be done much earlier. So in this study, we injected metastatic cancer cells intracardiacally, and they arrest in the brain almost immediately. So these were labeled with iron, which allows us to see them with MRI. So these are single cancer cells that have arrested in the brain. And we can see them both with MRI as well as MRI. And we wanted to do this because in MRI, the signal voids that you see with MRI could be representative of both dead and live cells that have arrested at this time point, whereas BLI we know is a measure of just viable cancer cells. And then similar to the previous study, we can also track these as they progress into metastases. We can see the hyperintensity in the MRI image as well as the viable tumor tissue from the BLI. So the other option is to take multiple images of the same signal by rotating the animal to better predict the origin of signal. So many people forget, but this is a very simple option that doesn't require any additional features or time. So here we see this animal. We can see there's a small signal on the animal's right side. It's on its back. And as we start to rotate and take images of this same animal, we start to see just past 90 degrees, we can see, okay, yeah, it's on the right side of the animal. It's also fairly shallow. So it's almost directly under the skin at the top of the, at the dorsal end of the animal. Similarly, we can see that there is this signal on the left side of the animal. but we don't know the depth. And so once we start rotating and taking pictures from different angles, we start to see, okay, this looks like it's probably in the hind limb. So you can see it's not a perfect science, but it definitely can really help you to better localize where your signal's coming from. The next option is to perform ex vivo imaging and take images at different levels during dissection until your signal is found. So this is a really great option, both for those doing fluorescence and bioluminescence imaging. And for those that are interested, just for sake of time, I'm not going to go into details of how to do ex vivo imaging, but for those that are interested, feel free to reach out, and I'm happy to help you because I know this can be a technique that some people are less familiar with. And then finally, you can use 3D tomography to co-register signal with an organ and bone library. So here I'm showing a feature that the Newton system has. I believe there are other 3D tomography systems out there. I'm not as familiar with them, but I encourage you guys to explore those systems if you are interested in this feature. The Newton system, for example, allows you within the analysis to load a organ library and bone library. So you can actually choose which organs and bones you want to show, and you can see where your signal falls within that. So I'm just going to fast forward to this slide so that we can get this video playing. You'll see the signal within the volume of your animal, and then slowly it will load in the organs and bones. So not super useful for a simple tumor model necessarily. So here it's just a mammary fat pad tumor, but you can imagine if you were looking for something maybe in the abdomen or the lungs where those organs are all packed in together, this can really be a great starting point to better figure out where your signal's coming from. How do I manage multiple signals within the same animal? So this is another great one. You'll see here I'm showing an example on the top where we have a mammary fat pad tumor. It's growing over time. It's very aggressive. You start to see that loss of signal in the center that we talked about earlier. Now, because this signal is so strong, what's happening is we're missing other signals. So what I encourage you to do, especially if you have something that you know is metastatic or you're not sure, but it's a very aggressive tumor model, cover it up when the signal is this strong. And what you'll see over time is that you're actually, you may have other signals that are not as strong that are getting confounded by this stronger signal. So covering it up will allow those to be detected. Multiplex imaging. So this is a great option for those of you that are looking at treatment studies or maybe tracking multiple cell lines within the same animal. It's basically the simultaneous acquisition of multiple signals within the same animal. So here we have an example where you can look at both red and green fluorescent signals, and then they're overlaid so that both reporters can be visualized simultaneously. So some of the common applications where this would be really useful, cell tracking. So as I mentioned, looking at maybe a cancer cell population that is red and a therapeutic cell that is green, being able to see the interaction of those populations within the same subject can be really beneficial. Secondly, immunology. So looking at various T cell populations potentially, or maybe a T cell population and a cancer population. And then finally, neurology. As the brain, we know the brain biology is very complex. So being able to look at more than one cell population can be really advantageous here, especially with only one modality needed. Okay, and then finally, I wanted to end with this slide because I think this is the most important. Help, my data looks weird. This happens to all of us. Definitely happens to those that are new to optical, and it really freaks people out. So I've done a lot of collaborations where maybe it's a group that doesn't typically do animal work, or they haven't done any imaging. They've spent 2 or 3 years on in vitro studies, and their data looks Really great, really clear results. They're super excited. They want to get that Nature paper. So let's just do one final imaging study to validate everything we've done in the last 2 or 3 years. We do the imaging study and they don't get anything like what they're expecting. And it contradicts all of their in vitro work. And people get really, turned away from this. And I think you really have to embrace the fact that, first of all, optical is a whole body imaging modality. I think that's a really great advantage. Some people like to tune into the specific region that they're interested in looking at. But I think it's important to know that there may be other things going on that maybe you were blind to before, whether you're using other imaging modalities that haven't been able to capture the whole field of view, or you haven't done imaging entirely. Understanding what your signal means, this is really important. So different, depending on the luciferase or substrate or reporter that you're using, your signal is going to mean different things. So for example, if you're using firefly luciferase, your substrate is going to require oxygen and ATP. So some people get really confused when their tumor is growing, their signal goes up, and then maybe after 2 or 3 weeks, their signal goes down. And they say, that doesn't make sense. The system must be broken. The signal's wrong. What's going on? Well, because your signal's representative at that point really of only viable tumor tissue, it makes sense because one, either your treatment is working, and even though your tumor volume may be going up, the amount of viable live tissue may be going down. That's a good thing. The other thing to consider is that if your tumor is getting really big, like I showed in the previous images, you may have a necrotic core, and that could also be causing your signal to decrease. It's important to remember that molecular changes occur before anatomical changes. And so again, the size of your tumor may not be representative of viable cells. Perform in vitro experiments to characterize your cell lines whenever possible. So you'll see here in the bottom right, at the very least, always try and do a very simple in vitro experiment to correlate the number of cells to the amount of signal. You should see a very nice linear correlation if those cells are stably expressing your reporter. I recommend doing this even if, let's say you're buying them from a supplier, they've done all of this, you've seen all the data, maybe you're collaborating with a lab and they've offered to give you this cell line and they've done the characterization. Always, always, always try and do it yourself in your own hands. Again, I know it's annoying, it takes time, it takes work, but it's worth it. It's going to work differently in your hands, and you don't want to inject 20 animals and find out, maybe you're using a different substrate than your collaborators, and for whatever reason, it's working differently. So always, always, always do some sort of in vitro characterization, even if it's just a simple cell number versus signal test. Finally, if all else fails and you've been doing the same study for months on end, you've tried all other avenues and your data is still looking weird, as a last resort, you can always use a luminometer device. So depending on where your optical system is from, you can always reach out to your manufacturer, the supplier, and see if they can run a quick calibration using a luminometer to make sure everything is working as expected. Okay, so in summary, most important thing to take away from this is be consistent. When I say this, I mean in terms of positioning your animal, reporting signal, copy and pasting your ROIs, substrates, where you buy your substrates, how you make them, how you aliquot them, always be consistent. Be prepared for variability. I think that's really important. Optical imaging, there's lots of variability. There's lots of confounding factors that can affect your signal. So always be prepared. Embrace signals you weren't looking for. This can be tough, but it's a good one. I think the best science often comes in places that we weren't expecting. So try to be open to that and try to always start with whole body imaging to see if there are signals that you weren't necessarily expecting. And then finally, always use the peak signal for quantification. And with that, I think I'll turn things over to Tanya for a quick poll question. Oh, Tanya, I think you already did the poll question, didn't
I
you?
did. Yeah, I was going to say
Sorry.
while we were sorting out your audio issues,
That's
we did
right. I
run the
totally missed
poll.
that.
No
Awesome.
worries. If anyone wants to have Katie reach out to talk more about the Newton system, just enter it in the chat if you didn't enter it in the poll question, and we'll make sure that Katie does reach out just to talk more about the system. But at this point, let's just jump straight into the Q&A session. So again, we have a few questions that have come through, but if you have other questions that you've thought of, please feel free to enter them in the Q&A dialog box, and I'll get started with Katie and we'll work our way through. So a question here, how do you choose the most appropriate reporter? For example, you mentioned a couple of different luciferases, so how do you know which one's the best?
Yeah, that's a great question. I would say the application is really important here. Otherwise, you need to consider how comfortable you are with the less explored luciferases. So if you're very new to optical, again, I would say try and stick with fluorescence just to start, or firefly luciferase. Those are the easiest to start with. They're also quite sensitive. But there are other things to consider. So, for example, I would definitely explore the cofactors that are required. So, if you're someone that's looking into doing a treatment study, I think making sure that you're choosing a luciferase where your signal is going to be representative of viable signal only. So, something that requires ATP and oxygen is important. You also want to consider the substrate delivery. So some luciferases, for example, Ranilla luciferase, the substrate needs to be administered IV. So as long as that's something that you're qualified to do, then I think that's pretty open. But other than that, I would say explore the literature and make sure that the luciferase substrate pair that you're choosing makes sense for your application.
Perfect. And if you're imaging more than one animal at a time, which I'm sure many people are, is it possible that an animal with a stronger signal could affect other signals in the same way that you discussed the two signals being a problem within the same animal? And how would you suggest dealing with this?
Yeah, so this one's tough because I think a lot of people explore optical imaging because it's so high throughput. So many systems now are 3 or 5, even 10 mouse systems. It's really tempting to throw 5 or 10 mice on there at once and get imaging data from all of them. I think this is definitely an option that's possible for those that have lower signals, or maybe near the beginning of your experiments. You just want to make sure that you're not saturating your signal. So if you have, let's say maybe you're imaging 5 mice at once, and you have one mouse that has really, really strong signal, and maybe you're not seeing signal from the other mice. I mean, your first reaction, you may think that maybe your substrate injection was not good, maybe your cells didn't grow, etc. My first response would be take that animal out that has the really strong signal and try imaging again and see if you can see those more faint signals without that strong signal in there. You can also try covering it. So similar to how I showed, if you have multiple signals within the same animal, you can try and do that where you cover up the animal to see if there is signal that you're missing within the other animals. But I would say if you have an aggressive tumor model and you know the signal is going to be stronger at later time points, try not to image them all at once, just to make your data as reliable as possible. It's probably important in that case to image them maybe 2 at a time or 1 at a time to make sure that you're getting all of the signal that you can possibly get.
Perfect. Another question here, how long should you wait to image after injecting your substrate?
Yeah, so this is really going to depend on the substrate you're using. For deluciferin, I find more often than not, people are injecting their substrate and then, getting things set up. Maybe they come back 15 minutes later to image because they think that it takes 15 minutes for the substrate to circulate. I would actually recommend injecting your substrate and putting your animals in the scanner right away. It's not going to hurt them by, collecting more images. It's relatively inexpensive. And you don't want to risk missing that peak. So yes, it's probably safe to say, if it's your fourth time imaging these animals and the peak usually happens around half an hour, it's probably safe to assume you're not going to miss it within the first 5 minutes. But you never know. As I mentioned, the peak can change as the disease model progresses. You also need to consider which luciferase substrate you're using. So, for example, ranitidine the peak can be, within the first minute. So in that case, I would even recommend putting your animals in the scanner, positioning them, having your acquisition parameters all set up, and then actually injecting them in the scanner so that you're able to image seconds after the injection.
Right, that makes sense. Again, anyone has questions, feel free to put them in the Q&A dialog box. We'll work through, let's go with one more question before we reach the end of the webinar. So on slide 14, you talked about a big tumor with a core in the middle. Do you feel that a low vasculature, so lack of blood or oxygen to the core of that tumor, also could have contributed to the lower signal and the ring structure that was observed?
Yeah, absolutely. And this is kind of what I meant when I said there's just so much variability. It could be a lot of things. This is when histology is really our best friend. It could be limited vasculature, especially with a tumor that is that aggressive. It could also be a necrotic core. And also that scabbing that we saw. I mean, you can visually see it. It's definitely, absorbing some of that light. So I think it's likely a combination of things. But your histology will really be able to, confirm whether the vasculature is playing a large role. But I predict, yes, it's absolutely probably playing a role.
Perfect. Excellent. So we've reached around the 60-minute mark for today's session. So I do want to be respectful of everyone's time. So we'll wrap things up. Any unanswered questions, we will either get back to you directly or put out a transcript over the next few days. I want to thank Katie for her presentation today, her expertise with the optical imaging and all the information that she shared with everyone today. I trust that we've been able to provide you with some relevant information that you can take back to the lab and hopefully implement into your studies. If in the days and weeks to come, you come up with other questions or thoughts and you want to talk further with Katie, we're happy to have you reach out to us here at Syntica Instrumentation. If you want to talk more about our Newton optical imaging system, I also encourage you to reach out. And those of you that answered accordingly in the poll questions, Katie will reach out over the next day or two as well. At any point, we welcome the opportunity to talk about your specific research goals and how any of the equipment that we have here at Syntica can help move that work forward. Thank you again for taking the time today to join the webinar, and we look forward to seeing you at another Syntica Instrumentation event. Have a great day.